Happy Saturday, scientists! Let's review. This week we begin the mixed bag of biology topics, B6 or global challenges. It has everything from studying ecosystems to studying health and disease. It's a long one, so we'll be splitting it into 2 videos. In this first part, we're going to cover the ecology sections, and in the second video, we'll talk about the health and disease sections. If you need to, remember that you can pause the video at any time and come back to it later. Revision is a marathon, not a sprint. Let's begin. We're starting by looking at a study of the world around us, and as you've probably noticed, it's a rather big and rather complicated place. Because of this, we have to rely on something known as sampling. That is to say, we take a sample or a small part of the overall ecosystem or of the number of individuals, and we study that part in detail and assume that the whole larger part is represented by that sample. We have a number of different sampling techniques that we use when we are trying to capture organisms, and we're going to start by going through those, what they're there for, and how we use them. The first technique is to capture insects, and it's known as a pooter. And it's a small device that has sort of a little glass or plastic container with 2 tubes coming out of it. And on one of the tubes, there's a small screen. That screen is there to block the bugs, so you've got yourself on the side there. And so what you're going to do is you're going to suck through the bugs, and then they'll end up in that small container and you can study them. The next technique is for capturing flying insects in the air around you, and that is a sweep net. So it's exactly what you think. It's a long net on a pole, and you sweep that through the air to capture insects that might be flying. Your next technique is kick sampling, and it's used to capture invertebrates in the riverbed. So if this is your river here, what you'll do is you'll disturb the bank or the bed there, and you'll have a net downstream that you can then capture your disturbed individuals in that net, and then you're able to study those. Tree beating is kind of exactly what it sounds, and what we'll do is we'll use it for capturing insects that are living in a tree. And so what you do is you put down a large white sheet so that you can see them, and then you put that down around a bush or a tree, and you hit the bush or tree with a stick. They all fall out, and then you're able to capture them, sometimes with a pooter or sometimes just looking at them on the sheet there. A pitfall trap is used to capture crawling insects, and it is what you think. You have a small hole that you dig in the ground, and then the insects will crawl into that. You do have to remember to cover the hole there so you don't have water that comes in. So your insects will crawl and fall down and be trapped. When you do any of these techniques, you need to make sure that you are treating organisms ethically. What this means is that you're not being cruel. You're not harming the individuals unnecessarily. You're not killing individuals unnecessarily. Once you've captured these individuals, you need to be able to identify them. And this also applies to plants as well. And it's usually done by means of a key. What you'll have is you'll have a series of branches or choices. For example, start with leaves, and you might have jagged leaves or smooth leaves. And then under jagged leaves, you might have some that are narrow, and you might have others that are wide. And then that narrow one will lead you to identify it as organism 1, and the wide ones will lead you to identify it as organism number 2, and it'd be the same underneath the smooth leaves. So you have a series of questions or series of branches, so that's why this is called a branch key. The other type of key that you have is called a dichotomous key. And the name comes from the fact that it's a series of questions or a series of dichotomies that you have to answer. So you'd start, if we're doing the same one, with jagged leaves or smooth leaves. And then that would lead you down to question 2, and the other one would lead you to question 3. And then question 2 would ask you narrow leaves, and that would tell you that it's organism 1, or wide leaves, and that would tell you that it's organism 2. And these can become much more complicated as you try and identify the various different individuals that you're dealing with. If you're making one of your own on an exam or test question, make sure you're choosing really obvious choices and kind of try out the key yourself to make sure that it works and you can't be led down the wrong path, for example, as you do it. The next thing we need to do in terms of our study of the world around us is to be able to estimate the population, i.e. to understand how many individuals there are. And it is nearly impossible, it's certainly impractical, shall we say, to count every single individual that exists. So what you have to do is you have to get an estimate. You have to get something that's close to the true number, but is not necessarily that true number. And these methods have been tested and have been proven to get fairly good results. So let's start by talking about how we do this for animals. The technique we use here is known as the capture-recapture, or sometimes known as capture-mark-recapture. And it kind of is what you think. So the first thing you do is you capture some animals, and this is done using either one of the techniques above or a different technique for larger animals, and this is known as your first sample. Then what you are going to do is you are going to mark these individuals. Then what you're going to do is you're going to mark the animals that you have captured. And you can do this with paint, you can do this by shaving a small patch of fur, you can do this in a number of different ways. Radio tags as well, actually. There's a whole bunch of them. And what this does is it has to be non-harmful. It can't make it more likely for the organism to be preyed upon, so you can't, paint them bright orange. It needs to be non-destructive and not have an impact on how the organism survives. Once you've done this, what you are going to do is you are going to release the animals, and then you are going to wait. And how long you wait depends on what animal you're dealing with. If it's slow moving, you might need larger time. The idea is that they remix with the population before we go in and recapture. Although you're getting here just a general new sample, and this we call your second sample. So really the word recapture isn't great here. It is a capture of a new sample that we're doing. Once you've got that, what you are going to do is use the formula here. So population is equal to the number in the first sample multiplied by the number in the second sample and divided by the recaptured. So if your first sample was 10, your second sample was 15, and you recaptured 6, then you'd have 10 times 15 divided by 6. Okay, let's move on to plants here. Plants are obviously non-mobile, so we're going to go about sampling them in a different way. We're going to use a technique called quadrat samples. In this technique, you've got these 1 meter by 1 meter squares, or usually they're 1 meter by 1 meter, and they've got a grid pattern laid on them that you can use for figuring out what's going on. And so your first step is to figure out your field. So if this is the field we're going to sample, we need to establish a set of coordinates on that field. And then what you're going to do is you are going to use a number generator, a random number generator, to get coordinates to that field. And that is what you're going to use to place the quadrat. So you'll get two random numbers and you'll place the quadrat at those coordinates. Then what you are going to do is you're going to count the individuals in the quadrat, so that each plant or each number of plants, so that you can see what's going on. So you'll count the individuals. Your next step is to repeat this. And you're going to do this a few different times depending on your resources. You want a good number of samples so that you get pretty close to the true population, but you also don't want to waste the time and resources that you have available for your study. And then once you're done that, you calculate a mean. Your next step is to figure out how many quadrats will fit on the field or in the area that you're sampling. So you're going to take the area of the field and you're going to divide it by the area of each quadrat. To use an example here, let's say that our field is 100 meters squared, and then our quadrat is 1 meter squared, so we would fit 100 quadrats on that field. So what we can do then is to use a formula that the population is going to be the average or the mean number of individuals in one quadrat multiplied by the number of quadrats on the field. So we're going to assume that every quadrat on the field has the same number of individuals, and so therefore that will give us the total population. We discussed briefly the concept of random and non-random sampling as we were talking about these. Random sampling is very good for avoiding bias, and so it's good for things like population samples or population studies, where you don't want to just be studying a certain area, you want to kind of get an idea of the total population size. Non-random samples, which are often referred to as transects, although you can still place the transect itself randomly, and you probably should to avoid studying certain areas. You use these for things like edge effects, or population distribution, so to see whether or not a given abiotic or non-living factor affects how the organisms live, you'd use a transect sample, and you'd use non-random sampling. So within the transect, you aren't being random. So remember, your transect is usually about a 1 meter by 10 meter grid. Sometimes they're a little longer or a little wider. And then what you do is you have that divided into sections, and you sample along the sections to see how the distribution of organisms changes along the sections. The next topic we are going to discuss is the concept of biodiversity. And what we mean here is we mean the number of individuals of each species that live in an area, and we mean the number of different species. So you need both of these things in order to have a biodiverse area. So you need lots of individuals of a lot of different species in order to say that an area is very biodiverse. Unfortunately, we now have a lot of things that are removing biodiversity or that are causing biodiversity to go down. in our world. One of these things is deforestation. And this has been done for the resources that are in the forest, but it is also being done more and more for farming, to create mass farms. And both of these things, what they're going to do is they're going to remove habitat. So all the organisms that live in the trees or around the trees no longer have a place to live, and they will die off or move elsewhere. And that'll mean that biodiversity goes down. We also have just agriculture itself. So not only does deforestation for agriculture cause a problem, but the monocultures that we tend to see in fields, or just one individual. So remember, number of species is important. If you're only growing one thing in your field, then by definition you've lowered biodiversity. You're also likely going to remove the hedgerows at the end of your field so you can get your machinery in there to be more efficient at farming. You will probably use herbicides and pesticides in a lot of cases. And then we've also got the obvious categories of hunting and fishing. If you are removing organisms from the environment, then you are decreasing biodiversity. And lastly, we've got pollution. So pollution can affect in two ways. It can directly kill off organisms, but it can also change the distribution of organisms. So if you kill all the plants in an area, then the animals will move away from that area because they need to move for their food. So this has led to a number of different ways of trying to conserve biodiversity, or at least keep it where it currently is. One of those ways is to use controlled grazing. So you can set out when individuals are able to graze, or when things like cow and sheep are able to graze in which fields. And we can also establish protected areas for them, so places where you can't hunt, or sometimes these protected areas are seasonal, so you can't hunt in mating season. In really bad years, we can feed the animals just generally or within those protected areas. We can also look at captive breeding programs, and these are usually done in zoos. So you'll have the individuals in the zoo breed, and then you can eventually reintroduce their offspring to the environment. The next thing we need is international agreements, because animals don't recognize borders, and so you need to have an agreement between organisms, between countries, to stop this from happening. So one example is CITES, which is the Convention on International Trade in Endangered Species. It makes that illegal. We can use seed banks to store seeds and to reintroduce them into the environment. We can also allow some limited ecotourism. And so what this means is that we can control the number of people who are going into an environment. And because those people will be paying to go into the environment, or paying individuals in the community, then we can use the money that they're paying to reinvest into the community, or into controlled grazing or protected areas or feeding animals, kind of reinvest it into those programs. It also means that the local economy will go up, and so people would be less likely to, for example, engage in poaching activities because they don't depend on that for income. So there are a number of benefits there as well. The next thing we'll talk about is feeding the human race. We're a growing population and we require more and more food each year. And so that has led to some innovations that have come about to allow us to try and produce more food. The first of these is selective breeding, which is where you breed individuals for their desired characteristics. And so the first step to this is to select individuals with high levels of the characteristic that you are trying to have, that you're trying to encourage. And you will then breed those individuals together. And once you've done that, you're going to get some offspring, and you will look at the offspring and find high levels of desired characteristic again, and then select individuals from that group and repeat the above steps. There is, however, some problems with this. We will limit the genetic variation here, right? So it's called a genetic bottleneck. And what this means is that you're narrowing the variation or limiting the variation in the population, and you're also limiting the gene pool. So this can create problems by making individuals more vulnerable to diseases. So we might have individuals that would otherwise have been resistant to a disease, but now they're more vulnerable. And so one tiny disease would wipe out your entire herd. This is also going to bring along undesirable characteristics. So if you think about it, because you're breeding the whole individual, not just the parts that you want, if that individual has some of the parts you want, but potentially also has some stuff you don't want, you're still going to bring those things along with it. So you need to be a bit careful there in how you select individuals. What you're doing is you're taking just the genes from one individual or one species and you're going to put them into another species. And what this will allow you to do is to create a transgenic organism. That means that you've got two different species that you've got together. And an example of this is the frost-resistant tomatoes that were created using genes from deep sea fishes. If you are on the higher syllabus, you need to know how we produce these genetically modified organisms, and it is something that we have looked at before. Usually, we're going to use either a virus or a bacteria in order to move the gene, so a vector here. And what we are able to do with either the virus is we're able to get the virus to inject its DNA into the cells of the host, because that is what viruses do normally. So what this means is we can give them the DNA we want and they'll inject that. We also do this via plasmids. So if you think back to what we've previously seen here with a plasmid, so that sort of circular bit of DNA in bacteria. And then we've got these two red bits here, which are to symbolize the sticky ends. And hopefully you remember that those are created by restriction enzymes, which are special enzymes that cut DNA at a given place. And so what you do when you're creating one of these, either with a plasmid or with a section of DNA in a virus, is you cut out using the same restriction enzyme your desired gene, and then those two sets of sticky ends will match. And so we'll have our desired gene, and then we'll have either our plasmid or just a random string of viral DNA, depending on what you're working on here. And you're going to use ligase to attach that together. So ligase is the enzyme that links DNA, and then you have a whole new plasmid that you can stick into a bacteria, which you will then get to infect the plant. Or if you're not using a plasmid, if you're just using a section of virus DNA, then you can stick it into the virus and it will infect the host cell. Sometimes you're able to do this with just the section of DNA that you want itself. So you can stick it into the little virus capsule there, and then the virus will attach itself to the cell, and that DNA will be injected in. So it depends on the application whether you use a bacteria or a virus, but you can kind of create these organisms. What this has led to is innovations in biotechnology. What this has led to is some biotechnology in farming. And one of the first ones that we came up with was herbicide-resistant crops. So what we could do here is we could spray a herbicide on the field and it wouldn't kill those crops, but it would kill everything else. And then we developed crops that could make their own pesticides. And this is the case with, for example, BT corn. It produces its own pesticide and it's able to kill off the things that try and eat it. We've also looked at increasing the value of a crop, or increasing the nutrition of a crop, and increasing the drought resistance too. So we've got drought resistant crops, and we've got these crops that have higher levels of nutrients. A great example of this is golden rice, which is high in vitamin A. I hope that you have found this useful. As always, if you have any questions, please ask those in the comments below and we'll do our best to respond. There's a worksheet of practice questions in the description and the answers to that will be out next week. Until then, happy sciencing!